Hey everyone, let's talk Kelvin cycle next. So the second step in our photosynthetic process, or the synthesis step of photosynthesis, is called the Kelvin cycle. The Kelvin cycle is found or performed in the stroma of the chloroplast. And within the Kelvin cycle, um, it's sometimes also referenced as the light independent reactions because we don't necessarily have to have light in order to perform it. But without light, we can't have some of the key components of ATP and NADPH, which drive the process of the Kelvin cycle. So it's kind of a hand in hand piece that goes along with that. So the Kelvin cycle, again, it's called the synthesis portion because carbon dioxide is going to go through this cycle in order to create or make our glucose molecule, which is the end product and end game of our photosynthesis process. So the Kelvin cycle, again, it's three main components here: ATP, our energy rich molecule, NADPH, also energy rich like ATP, and then the carbon dioxide molecule is also going to go through and cycle through the Kelvin cycle. There are 3 steps. There's always 3 steps for every scientific biology phase. I don't know why, but it kind of makes things simple. So if you're ever going through, you should be able to say, hey, there's 3. It's called carbon fixation, or basically when carbon attaches and gets linked in with another molecule. There's the reduction phase. So that means we're going to lose something, right? Reduce or decrease the amount of something. And then there's the regeneration phase. So making something that was there at the beginning of the cycle that was no longer there. So then the cycle can continue. Fixation, reduction, regeneration. Those are our 3 steps of the Kelvin cycle. So, all right, step 1, carbon fixation. And you don't have to memorize the steps, but we should be able to address, ATP, NADPH, and carbon dioxide and how those are utilized to make sugar. So for example, in step 1 of carbon fixation here, we're going to capture or combine and fix carbon dioxide to a molecule, which is called RuBP, or that's the shorthand for it, but it's called ribulose biphosphate. Again, you don't have to know that it's called that. Okay, so we just call it RuBP for short. But we're focusing on the fact that carbon dioxide is going to be combined in order to make this larger molecule, and it's called a 5C or a 5-carbon compound called RuBP. So here's my first step. In comes carbon dioxide into this arrow here, right? It's going to enter in and join up and hook up to create this RuBP molecule, or 1, 2, 3, 4, 5. Why do we care about this? RuBP is going to be a substrate that combines, right? And then we can see, and it's going to create this little Rubisco enzyme, which is really, really important in processing carbon dioxide. It just speeds it up really, really fast. So that's an important enzyme that's involved here. So, and there we go. This was one of the molecules that I referenced way, way back in our biochem section that was really important to look at. So if you ever research Rubisco, you'll see that it's the most abundant protein on Earth and super important. Okay. All right. So that combination will then break up those molecules into what are called PGA molecules. Do we care what PGA stands for? No, we don't. But just know we'll go from CO2 to create a RuBP, and then that RuBP will be split into PGA molecules, and that's just kind of what's happening over here in our process. And then this happens 3 times. So we can see at the beginning here, input, there are going to be 3 carbon dioxide molecules that enter in and go through this fixation process. So by the end of it, we're going to end up with 6 3-carbon PGA molecules. And really, we're following the process of carbon because that's how we end up with our glucose molecule. And that's the important step. So having those 3 carbon dioxide and carbon dioxides enter, and then we end up with 6 3-carbon molecules. Those are going to be key for our next step. So that's carbon fixation. The next step is reduction. So over here, we're going to start seeing the reduction part. This is where ATP and NADPH, those energy-rich molecules, are going to come on in and start driving this process of rearranging our PGA molecules. So it looks like we end up at the end of this reduction phase with kind of like a half a sugar, which is going to be kind of important. So again, there's lots of steps. There's crazy names that go with them. We don't care about their crazy names. We just are able to say, hey, ATP goes through and cycles, NADPH goes through and adds its energy and helps cycle, and then reduces that step so that one of these PGA molecules is then lost and passed on to be stored and saved later so that we can make a glucose molecule in the end. So that's what's happening over here. One of our molecules is dropping out so that it can be used later on as sugar or other organic compounds. It's reduced or lost. And then our third step is called regeneration. So after the reduction process, we need Ruby P again. So it's going to go back through our remaining 5 molecules are going to go back through. ATP is going to drop off some energy, drive it up to recreate this Ruby P molecule. That is going to then go back through and restart the first step or the first phase of the Calvin cycle. And this is going to happen a second time so that when we go through this step and we get back to the reduction phase, we have our second half of our glucose molecule or our organic compounds that are necessary for life. So this cycle, this Calvin cycle, has to go through twice in order to make 1 glucose molecule. So if there are initially 3 carbon dioxides entering in, we go through it twice, so there's going to be 6 total carbon dioxide molecules that go through. And if we have 6 carbon molecules, that's important because in the end, that's where our glucose gets its 6 carbon, atoms. I think I said molecules earlier. The 6 atoms of carbon will be utilized for glucose later on. So again, just remember their phases, or the phases, right? Fixation, reduction, regeneration. During fixation, carbon dioxide is combined in order to create a Rubisco molecule, but we don't really care about that. Carbon dioxide is entering and making a larger molecule. During the reduction phase, that larger molecule is going to be broken down by ATP and NADPH so that one of those molecules can leave and be half of the glucose molecule over here waiting. Then during the regeneration phase, ATP can go through and recreate a Ruby P molecule. So then we can go through the Calvin cycle a second time and finish off the fixation, reduction, and then we have our other half of our full glucose molecule over here, and regeneration. And it can just keep on going as long as there's ATP, NADPH, and carbon dioxide present.